Alright, so this is the pre-lab now for fungus. It's the last pre-lab for Unit 2. Alright, so for fungi. Fungi now we're still in the domain eukarya. We are now then into the last part for your exam for Unit 2. So keep in mind then Unit 2's exam is going to cover virus, archaea, bacteria, which is prokaryote. Then you're also going to cover protista and fungi, big chunk of life, so I really wouldn't, put this off till last minute because it's a lot. You're also going to have a lab practical too. It's going to be based upon, what I'm having you do in the labs anyway. It's two separate grades. The exam grade, you understand, is kind of like your Unit 1. It's a standalone grade. The lab practical grades, whatever you get on the first lab practical, I add it to the next lab practical, to the next one, to the next one. I add all four up, and then hopefully it's a grade that you're happy with. All right, but that won't be until, let's see, next week. That won't be for a little while yet. All right, so for fungi. Fungi, understand now, unlike the protists, they are very much more aligned. So with this group now, we're looking at major traits. They're going to have cell walls. Their cell walls are going to be made of chitin. That's how you say C-H-I-T-I-N. It's not chitin, it's chitin. So it's a different form of outside covering. So keep in mind, keep these in your head. For bacteria, they had a cell wall made of peptidoglycan, protein sugar. For archaeans, they had a cell wall made of pseudomurin, different version. For fungi, cell wall made of fungi. Cell wall made of chitin. When we get to Viridiplantae, cell wall made of cellulose. When we get to animals, no cell wall. That is a way to classify. Just start keeping these things, in your head and keeping track of them. All right. So for fungi, they are heterotrophs. Yes, they're consumers. However, what you have to understand is they are what are called absorptive heterotrophs. Absorptive means they'll start digestion outside of their fungal body and finish it off on the inside. Their fungal bodies are made up of these little tiny strands called hyphae and mycelium, and they'll release digestive enzymes into the surrounding environment. They'll start to break that down like a straw. They'll suck it back in and finish it off. You are not an absorptive heterotroph. You don't take your hamburger, you don't vomit on it, you don't wait 20 minutes and eat it up. And if you do,
Ew.
Don't tell me that, because that's just disgusting. What you do, you're an ingestive heterotroph. You chew it up, you swallow it, then you break it down. You don't break it down outside first. All right, along with this too, keep in mind that for fungi, everybody's going to be multicellular, except for one small group, yeast, which Saccharomyces cerevisiae is also in the lab because we have to pay homage to the one that is, unicellular in the group. All right, now, along with this, there's going to be, again, several phyla underneath here. So I'm going to go from the most primitive to the most recent, we'll say. Okay, so we have Chytridiomycota. The root word, notice, is myco. Myco is for fungus. So Chytridiomycota, they're going to be unique because they're going to be the only fungal group which is going to be aquatic. Then we're going to go into everything else from here on out is going to be on land. So we have Zygomycota, the most primitive of the terrestrial fungi. That's like bread mold, fruit mold. Then we go into another group called Glomeromycota. Glomeromycota is the smallest group in fungi. Environmentally, ecologically, one of the most important ones. These are the ones that form, it's a symbiotic relationship on the roots of the majority of plants on planet Earth so that they actually can grow faster. It increases the surface area for them to absorb water and minerals. So therefore, they're a very, very important good fungus in the environment, or the Glomeromycotas. Then we have the two big categories. First big category is going to be Ascomycota, also known as the sac fungi. This is going to be everything from yeast to truffles to morels, and then bracket fungi, which is going to be the one, sorry, not bracket, cup fungi. Cup fungi, which is the one you're going to see in lab. Then we come to the group, which is probably what you're most familiar with when you think of fungi, mushrooms. That would be then Basidiomycota. They're called the club fungi. So you have toadstools, mushrooms, puffballs, bracket fungi, jelly fungi, rust smuts. They're going to be parasitic. Then we come to the last group, which is going to be called Deuteromycetes. Deuteromycetes, again, notice each one of these has the root word myco somewhere in it. Deuteromycetes used to be part of the big group, Ascomycota. Again, with genetic testing, discovered that they were too different from Ascomycota, and therefore they had to become their own category. Two notable ones in there are going to be Aspergillus, which is what makes soy sauce and soy paste, and Penicillium, which is where penicillin comes from. Now, understand, in general, fungi are going to have same types of features to them. They're going to have little strings going throughout them. That's going to be called the hyphae. All the hyphae together is called the mycelium. That's the fungal body. Whenever they go to reproduce, they can reproduce spores either asexually or sexually. When they make spores, you got to be careful. If you have what is called the spore itself, sporangia, and the sporangiophore. When you have the suffix on it, a "phore," you're essentially saying, I am the structure which leads to the creation of. So if you're a sporangiophore, you are the structure which leads to the creation of the spore. Okay? Sporangium are the spores. Now that's a generic term. Because Ascomycota is so old and so big, these were called different names and they were grandfathered in. So when I say sporangia, when we talk about Ascomycota, we got to use the word conidia. Same thing. When I say sporangiophore in all the other categories, you say conidiophore in Ascomycota, again, because the term is grandfathered in. Because Deuteromycetes came from Ascomycota, those terms get grandfathered in with them. The reason why Deuteromycetes, it's also called the imperfect fungi. Why? Everything I'm talking about, where you're making spores and everything, that's common throughout all these categories. The hyphae, that's all common. So how the hell do you decide which category you're in? Most important thing about fungi, how we classify them is based upon how they make a sexual spore. So everything boils down to sexual spore. If you cannot make a sexual spore, that makes you unique. Like Glomeromycota doesn't make a sexual spore. Therefore, they've only been noticed to do asexual, so they're in their own category. The reason why Deuteromycetes broke off of Ascomycota, they don't form a sexual spore. So if they don't form a sexual spore, how the hell can you keep them in the same category as ones that make a sexual spore? So you make a new category. Now, the name of the sexual spores, if you ever get this wrong on tests, I should string you up. Why? Think. One group is going to be called Zygomycota, bread molds, bread spores. Its sexual spore is called a zygospore. Zygomycota makes zygospores. That's the redeeming trait. Cool. Ascomycota makes ascospores. Basidiomycota makes basidiospores. Correct. Chytridiomycota, the first one, is because it's aquatic, is unique. It's going to make zoospores. If you look back to what you drew for protists underneath saprolegnia, that was the one that if you don't when you wash your clothes and don't dry them, it smells funky. They also make zoospores as well too, so they were aquatic. Now again, back in my day, years ago, Oomycota and the slime molds were the first parts of fungi. Now with phylogenetics, they're more genetically similar to protists than they are to other fungi, which is why those two categories got pushed back to protists, and we start off fungi with Chytridiomycota now. Alright, so for your pre-lab, trying to think of anything else. Oh, also to keep in mind that fungi can also have these small little root-like structures coming off of them. They're not true roots, because when we get to plants, roots are meant to anchor the creature down, plus also absorb water and minerals. What these creatures are going to have are called rhizoids, which means, quote, root-like. They anchor you down, but they're not the greatest anchors, but they don't absorb anything, because all the hyphae absorb everything. So it's not a specialized structure, not a true root. Alright, so, anything else I need to mention about fungi before we get started? No. Alright, so, for the pre-lab. Alright, so, again, much smaller than the last lab on protists. So, first group then you're going to look at is Chytridiomycota. Chytridiomycota are infamous out there because they create a disease called chytridiomycosis. It's an infection you'll see in amphibians like salamanders and frogs. Basically, been wiping them off planet Earth since the 90s. So they're infamous because, again, think where a frog or a salamander live. Damp, dark environment. Think of the growing conditions for a fungus. It has to be damp, has to be dark, has to be cool, and there has to be a carbon source. Well, that's where a frog would live, the frog providing the carbon source, so would a salamander. Also explains why fungi like your fridge, because it's the same condition type of thing. All right, bacteria have the same traits, except for usually instead of liking it cool, the majority of them like it warm, which is why they like your body, because you're a nice little incubating oven. All right, so first one underneath in your fungi lab, chytridiomycosis, is Urophilictis. Okay. Urophilictis is an aquatic fungus. So what you'll see are like these little strands going everywhere, all around on the slide that I show you. Okay. Somewhere in it, then there'll be a clearing in the middle of it. Each little strand is a hyphae. The entire collection, again, is the body, the mycelium. Okay? Now, in the middle, then, you will see these little tiny circles. Usually they kind of have like a yellow tint to them, kind of like they're dried mucus, but they're just basically a spore covering is all they are. Inside of them, though, you'll see these little tiny cells. They're usually stained red, and you can see one little flagellum coming out of them. Those are called zoospores. Zoospores are important. Why? Because this is the only aquatic fungus, and why they're put in the categories because they make aquatic spores. Zoospores. Urophilictis. All right. So, next category we go to is then zygomycota. Zygomycota, if you remember, was bread mold and fruit mold, what I was saying. So, we're going to look at Rhizopus. Rhizopus is common bread mold. All right. So, with this, on the same slide, you're going to see both, again, all these fungi. If you remember back when I went over the seaweeds, I said you're going to go through alternation of generations. Gametophyte, sporophyte. Sporophyte is 2N, gametophyte is 1N. When you spend the majority of your life cycle in gametophyte, you are more primitive. Fungi spend the majority of their time as the gametophyte generation, very little as the sporophyte, so they're a more primitive creature. Now, in Rhizopus, the reason why I mention this is that you're going to see both sporophyte and gametophyte in the same picture at the same time. All right, so for sporophyte, if you remember, is diploid, full set of DNA. So what you would see is a lollipop-like structure, a stalk coming up, and then you'll see a ball on the end of it that kind of has this fingernail-looking thing inside of it, okay? This structure here is called the sporangiophore, meaning the structure which leads to the creation of the spores on top. This area up here, that kind of looks like a fingernail, is called a columella. The columella is where the spores bud off. And then what you see up here, it looks like a bunch of little dots, almost like a gumball machine, inside of a case. Those are the sporangia. Or sporangium is one, Latin sporangia is plural, all of them, okay? That would be the sporophyte. Nearby, what you would then see is kind of like a tube coming in, and a tube coming in, and this might be ripped apart, so you might only see this part here. But what you would see is this big, huge nugget in the middle of it that kind of looks like a raspberry almost in the middle. That is the gametophyte generation. This is haploid. That is the zygospore. Zygospore is where the name comes from, zygosporangia. That's the important one. That's the sexual spore. All right. Next slide is then Ascomycota. Ascomycota, if you remember, is the big sucker. Over 45,000 species in this group right now, currently. Now, in this group, again, we're going to have things like yeast. You're going to have cup fungi, which I'm going to show two different versions of cup fungi. Yeast, whenever you look at them, Saccharomyces cerevisiae, people see yeast and they see this little pellet, like, oh, that's yeast, sort of. That's what's called lipolysized. It's been freeze-dried with a oil of fat around it. When you add water to it, it reanimates. And when they reanimate, they're hungry, which is why when you make bread, there's always a small amount of sugar in it, but when you eat the bread, you don't taste any sugar, right, because you're feeding the yeast. When they digest that, they do alcoholic fermentation, they make gas bubbles, and your bread rises from the gas bubbles. That's why you let it set in a warm area, let it rest, and then the gas is given off and it expands, making the little holes in the bread. All right, it's also why beer has bubbles, champagne has bubbles. All right, now, with that, yeast is real simple. Saccharomyces cerevisiae, it's going to look like little tiny golden beads. Each one is a separate yeast cell. Nothing crazy about that. The only reason why it's in your lab, because it is the only unicellular representative in the entire kingdom, making it automatically unique. But it is an Ascomycota. All right, now, the one I want you to spend more time with is called Peziza. Peziza is a cup fungi. So if you were to see the whole fungus, now understand, these things grow in your yard right now. They're probably by your air conditioner. They're only about that tall. They're tiny little things, okay? So what you would see is this structure coming up that looks like this, and if you cut it lengthwise, it looks like a big smiley face on a stick. Now, if you were to take the whole thing out, you'd have these like little root-like structures coming off of it Those are what I said earlier, rhizoids. This little connecting area is called a stipe, which means a stem-like structure. Root-like structure, stem-like structure. What would you look at in the stipe if you cut it? You would see mycelium. Looks like mats of wet hair is what it looks like. If you look inside the smiley face, what would you see? Mats of wet hair, mycelium. What I'd like you to look at in the middle is if I took this area in the cup itself, if I take this area and blow it up, the smiley face, what you would see are these little finger-like bubbles, one after another. There's thousands and thousands of these things, all lined up, okay? The little bubble is called an ascus. It is the sac of sac fungi. Inside of the ascus, there will be 8 little, look like unpopped popcorn kernels. 1, 2, 3, 4, 5, 6, 7, 8. Looks like peas in a pod. Each one of those is called an ascospore. Hence the name Ascomycota. Okay? But that's going to be found on the inside of the cup. All right? That was Peziza. Now, Erifce. Erifce is a cousin. The picture that is taken for you is not at the angle looking at it as a longitudinal cut, cross-section type of thing. Take this same idea. Imagine, okay, so if you look at my pen lengthwise, you can see it's a pen, right? If I now take the pen and I tilt it towards you, what's it look like? A dot, all right? Same idea. When you're looking at the first slide, Erifce, you're seeing it like this. Sorry, for Peziza, you're seeing it like this. When I go to the next slide, Erifce, the same structure, this ascus, you're not looking at it like this, you're looking at it like that. Like down the barrel of a gun is what you're looking at, okay? So, when you go to Erifce, what you would see is just like mats of like little fibers everywhere. Again, that's the mycelium, that's the hyphae. Every so often, what you'll see is a dark ball scattered around on the inside. Well, what's the dark ball? You're looking down the tube of an ascus. Remember, the ascus has 8 ascospores coming down it. So it's kind of like looking down a pea shooter that has 8 BBs in it. You can't see all the BBs because they block the light coming through, so it looks dark. All you can see is the ascus. You can see the tube. All right? Now, a lichen. Yes, I know it looks like it's spelled like lichen, L-I-C-H-E-N. It's a lichen. Lichens grow all over the place in Florida. You got ones which are foliose, which look leafy. You got ones which are fruticose, which look bushy. You got ones that look crustose, which looks like paint on a trunk. Usually, if you go outside and look on any live oak, you'll be able to see all 3 on that live oak at different areas. But lichens, understand, are going to be symbionts to that plant. They're helpful. What they do is, again, they're going to have exchange of nutrients along with that plant. If you go back in your notes to when you looked at industrial melanism, that's whenever the lichens on the birch tree became dark because of environmental pollutants, because of the industrial revolution, because of coal soot. They are very susceptible to environmental pollutants, so therefore they're an environmental indicator for a lot of cases. Now, lichens, again, are going to be a symbiotic relationship between a fungus and either an alga or a cyanobacterium, the first lab you did with me with bacteria. Now, all I want you to be able to really see whenever you look at the lichen is what part is the fungus, what part is the symbiont. So, again, anything that looks like hair. On this slide, if I remember right, the hair looks like hair, but it's kind of stained almost like if I remember right, like a sea green color to it. So it looks like, just hair everywhere. That is the fungus. That's the hyphae, that's the mycelium. Then what you would see intertangled in the hair mat, you would see these little tiny balls entangled in it. That's the symbiont. That's the cyanobacterium. That's all I want you to label for that, to be able to understand for that one. Now we come to the group you're most familiar with, which is Basidiomycota. Basidiomycota are the club fungi, meaning things like mushrooms, toadstools, things like that, puffballs. Now, Coprinus. Coprinus is a regular, mushroom you would see growing in your yard, okay? So just think, when you see a mushroom growing in your yard, I'm sure you've all seen them before. Again, you got that stem-like structure. That's the stipe. You got the little rhizoids coming out. And then you'd have a little mushroom cap on it, right? So that part is called the cap. When it's not ready yet and it's still closed up, there'll be this white tissue underneath it. That white tissue underneath it is called the veil. Think of like a bridal veil. It covers it up. Once it matures like an umbrella, it pops open. And when it pops open underneath the veil, you'll see row after row of these little brown, looks like sheets of paper. Those are the gills. The gills are what house the basidiospores, the spores for it. Okay? So, when you look at Coprinus, imagine looking at a mushroom cap, and I've cut it. And you're looking at it cross-section. So what you would see are these long finger-like projections coming out. Like that, okay? This middle part right here, that's the stipe. Each one of these folds you see coming out is a separate gill coming out, okay? Now, what I want you, now again, if you looked at this and the gill itself, what would you see? Little hairs going everywhere. Again, hyphae mycelium. What I want you to look at, though, is on the gill itself, you'll see these little tiny bubble-like projections come out, one after another, and at the end of each bubble-like projection, there's a little black ball. Well, the little bubble is the basidium, and the little ball on top of it is the basidiospore. Why is it called a club? Because if you got it on both sides, it kind of looks like a club. You're going to whack somebody in the head, okay? That's why, all right? But that would be the basidiospore, and then underneath it, the bubble is the basidium. Then you'd have the gill underneath it. All right, now for Puccinia. Puccinia, same idea. I'm going to take the mushroom cap and cut it. But it's going to have a different format. Now it's going to have kind of like that going around, okay? Now what you do, again, here would be the cap itself, and you'd have the mycelium and yada, 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 all that. Inside here, though, what you would see is like a little stalk come up, and it looks like a ball on top of it. It's almost like a nut or a seed on top of it. Again, basidiospore, basidium, the club of club fungi. That would be then for Puccinia. All right, last two then. Notice I don't have any for Glomeromycota because I can't find anything that I like for that. So Deuteromycetes. Remember, this is a break-off group from Ascomycota. Deuteromycetes has no sexual reproduction, so therefore it's all going to be asexual. We can't use the words sporangiophore and sporangium because it came from Ascomycota. We got to use the grandfather terms conidiophore and conidia to mean the same things. All right, so for penicillium, aspergillus is first. Aspergillus, think of like when you're a little kid and you take a dandelion and you blow off all the fuzz off of it. Think what looks like when it's left, just the stem coming up with a little ball on it. That's what the fruiting body of aspergillus is called. By the way, fruiting body in fungi means anything that creates reproductive tissue, either asexual or sexual. It's the fruit of the fungus. So you'll have this stalk come up, and then it will look just like a spiked ball on top of it. Okay, and that would be then for aspergillus. This would be the conidiophore, the structure which leads to the creation of the conidia. Those are the spores. Okay? And again, when you look at it, you'll see all the little, you'll see the mycelium and the hyphae all around. I'm just pointing out the reproductive structures when I do this. Last is penicillium. Penicillium, again, a lot of this, when you look at slides, when they're dense, it's just big mats, and you can't tell what's what when it's all that dense. You have to go to the outside edges where it thins out, then you can see it. For penicillium, think about when you think of like wheat when it goes to make seed, or like your grass in your yard, if it grows too tall, make seed, kind of looks like that. So you'll have these stalks come out, and then they'll kind of just, flail on the end. In some cases, it almost looks like, looks like somebody's like a strange x-ray of somebody's hand. Okay, so this whole structure coming out is the conidiophore. When you look at the conidiophore, it looks like these little black seeds are in the pattern of the conidiophore. Those little black seeds are the conidia. Well, I'll do conidium. That's one. If I do more than one, it's conidia. Okay? And then I also have you look to label a mushroom, which I already labeled for you. And that should do it. All right, so next time I'll see you will be in Unit 2 for Plant Lab number 1.